this overlap. Okay. Thanks for coming. This is the BSCI353 Principles of Neuroscience. So if you're not taking that course, you're in the wrong place. I'll find somewhere else. Okay. This is actually a nice lecture, mostly a nice lecture theatre because it's a nice sort of amphitheatre and everybody is relatively close. It's a bit difficult because the people here, I'm up here and you're down there. If you want to vacate the first few rows and move a little further back, please feel free. I won't be insulted by that. Let's start off just with some mechanics here. So, firstly, this is a new course in the sense that we used to offer a course called BSCI353, Cellular Neurophysiology, and at the 400 level. And that course is being offered for the last time this semester. So for this semester only, we have this overlap. And the principal requirements for that course, prereqs, were BSCI330, cell biology, and physics 122, I think, the physics course. Those were the main ones that you sort of needed to have for that one. And several things happened to that course over the years. What really happened is that neuroscience has become a much larger field, much bigger than when that course was envisaged or was actually created maybe 20 years ago, 15 years ago. And so what our faculty wanted to do, especially our junior faculty and new faculty wanted to do, is to be able to deliver more specialized courses at the 400 level to those people who wanted to learn, really go into depth in neuroscience. And of course, if the first course that's called a neuroscience course in the sequence is already at the 400 level, then that really isn't going to work too well. You can't make that course a prereq, a 400 level course a prereq for other 400 level courses. Administrators don't like it. All right? So what we've tried to do, and the other thing that happened is the course was becoming increasingly a survey. Because as neuroscience grew and you wanted to explain a little bit about all of the new aspects to people, so the depth that one was working at decreased somewhat in any one aspect. And personally, I know that there are many 400 level courses that are survey courses that cover a lot of breadth, as it were, in a subject. But personally, I think that's not ideal in a research university. And so what we decided to do was to rename the course BSCI 353 to change the prereqs so that you could take BSCI 207, the sort of physiology survey course, as a prereq, as well as, or instead of 330, and make the physics course a co-req, if needed. And this coincides with a big push to try and produce a physics for biologists curriculum, which is underway at the moment, and to get people to take the physics earlier. Okay, so we're kind of trying to move things along by taking a large cohort of students at the 300 level, giving them a great course, and having them tell their people who come after them, this is a great course. Make sure you do the physics prereq before you take it. And we'll try and make it painless if you haven't, but we'd like to try and cram things down, right? And after all, it should be at a university that you're pushing material downwards, so that you're learning about new stuff at an earlier level in your career. If we were working in the other direction, well, that wouldn't be good at all. Right? You'd wind up not learning about the cutting edge stuff until later in your career. So by moving this course from the 400 to 300 level, it's natural. We think. Any questions about that sort of blurb or sequence or about the course? Okay. Somebody asked me online whether this will count towards the the Neuroscience minor in the same way as the 400 level course did. And my understanding is, I haven't gotten an answer yet from the two people who run that minor, but I'm pretty sure that it will do. I'm very confident that it will do. Because it was intended just to replace 453, both in terms of your graduation requirement from PhD and also, therefore, by extension, I'm sure, for neurophysiology. So I'll argue that case, but I don't think I need to. I think it'll do it automatically. But the sheet of courses hasn't been updated in at least a year, so I have to get on to Catherine and Cindy Moss about that. So that's one question I had. And then there are other questions about it. Okay, yeah?
How will the content differ from 453, if at all?
There's a section on computational neuroscience, which is new. And we're going to have to go a little slower when we work on the physics, some of the electrical circuit work. Apart from that, really, it's very much up to you to shape the content, in the sense that I think everybody was very happy in terms of students with the content of 453. And if we can cover that content at that same level, I think that would be a very good outcome. Maybe not the best, but it would be a good outcome. But it's very much up to you guys to work with me as we talk and to ask questions if you haven't learned about concepts in the past and you want explanations, to work with me so that we can mold the content so that it works at this 300 level. We'll also, I think, be doing a little bit more work. When I personally taught the cellular neurophysiology course, I spent a lot of time on the cellular stuff at the beginning and gave fairly short shrift to like the last 6 lectures, which are on things like emotions, sleep, attention, and things like that. And so I think I have a much stronger feeling of duty not to do that, because it's a principles of neuroscience course. Yeah. And if we go into cellular details too much, because we're still following that old syllabus, again, let me know. Right, this is an evolving thing. I mean, this class, I'm sure, is largely people who were perhaps expecting to take the 400 level class. Now it's been switched. But the future is not necessarily like that. Okay, any other questions about the material? Okay, so there's a syllabus. Everybody get online? Yeah, by and large, there's a syllabus online. I will put questions up as we go. You'll notice that there are just 3 exams. We're not doing any coursework, quizzes or papers this time around, because I'm trying to keep things simple and just concentrate on the syllabus and the material. So there are these exams, and the final is cumulative, so please bear that in mind. So objectives. So let's start with what the objectives of neuroscience is, because neuroscience is a sort of composite subject. It's not one of the basic disciplines yet. It's comprised of contributions from many areas, and in fact some of the most fundamental contributions in neuroscience have been made by people who weren't trained as neuroscientists. That's partly because it's a young discipline. So neuroscience, obviously, the objectives of the course are to understand the cellular and molecular basis of neuroscience, right? This is a biology course, BSCI, and so we will be concentrating on trying to relate neuroscience events in the brain and outcomes to the cellular, molecular, and structural level, wherever possible, evolutionary level. We will, by the end of the course, I think, understand how signals are generated in the nervous system and, in general, how they're processed. What are the basic building blocks by which the signals are processed? I think Also, there'll be several examples of how the nervous system can be modified by experience, which is a very important part of the nervous system, its function. And also, it's something that we've begun to get a handle on at the structural and molecular level. And then something that isn't happening, well, I did have one question, is it's up to you to ask questions. Right? I can stand here and rabbit on as much as I like, but you need to ask questions. And asking questions means, to some extent, reading the textbook or reading the notes beforehand. Yeah? You're all juniors and seniors. I'm not going to give you quizzes to make sure you do that. I'm not going to, make you do homework. All right? We're beyond that, right? Either you're going to do this because you want to do it, or not. It's all the same to me. Yeah? So do it because you want to do it, and ask questions, particularly if something that's said you don't agree with, or you'd like to know more about it, or anything, or you didn't really get. I would like you all to be able to leave the lecture with a good understanding of what was said in lecture, so that you don't have to spend a lot of time underlining your notes. That's my goal. Okay? Very efficient. Get it all done here and explained. We can all go outside and enjoy the sunshine. Yeah, I know, you're going to have it. Okay. But it certainly won't happen if we don't get some interaction. So I'm going to be asking you questions too, and I'm not shy to pick people out. One thing about me is if you ask a question and it's just off base, I'm a scientist. I go for the jugular. I say, that's the silliest thing I've ever heard, But I just can't remember faces very well. I'm not great at faces. I can can't link names with faces. That's my problem. So feel free. Say what you like. Say whatever you like. When you like it, right? Don't be disturbed if my reaction is, oh my God, how could she believe that? How could they believe that? Don't be disturbed by that, because I will not remember who asked the question by the end of the lecture, right? You can also put stuff on the discussion board. The rules there are, you may post anonymously. A lot of people do. But if you answer a question, and you're also welcome to answer your fellow students' questions. It's a discussion board, right? I know they're questions for me, but if you know the answer, dive in, I'll double check. And if you're wrong, you're wrong. If you're right, the answer, you're right, okay? But if you answer a question, another student's question, you have to sign it, okay? Because this is a science, right? And in science, we stand by what we say, what we think. And if I go to a meeting and I say something that's really daft, people are not shy to tell me, and my name is associated with that. Okay, so you're always welcome to ask questions anonymously, but if you want to answer somebody else's questions, you can still do it anonymously. I'm not going to rant and stop the email, but please, I would prefer it. Just put your name up. And again, I guess over the course of the semester we'll get to know each other, but basically, don't worry about my remembering your name and thinking, I've seen 5 emails from that person, and I don't go and hunt up your picture on eBay either. Okay, so we keep that very true. So what are the components of neuroscience? Molecular and cell biology, anatomy, physiology, which has a huge component these days of advanced imaging technology. All right? It's a big thing in neuroscience. Relatively new discipline is computational neuroscience, which of course comes from the fact that the brain functions as a computational device. It is processing information. And so many aspects of information theory, the idea, for instance, of bandwidth and frequencies, of discharge, all bit rates, all can be applied to neurons, computational neuroscience. Behavior, biologists call this neuroethology, right? Psychologists call it psychology in some cases, and those of us in the middle sort of call it behavior. Okay? But it's the output of the nervous system. really. Cognitive neuroscience is an area that I'm not familiar with, and I always get into trouble because no matter how I try and present stuff, material that is that I view as cognitive, I'm always told that it's not cognitive. It's basically biology or cellular neuroscience, pretending to be cognitive neuroscience. So that's an aspect which we won't be covering much of, and don't confuse cognitive neuroscience with sort of the kind of speculations that we have as biologists about how the brain works at a high level. Right? It's not the same thing. It's a separate discipline. And in evolutionary biology, it's important to understand the brain and the nervous system in terms of its evolution. It's been enormously helpful in biology to do that. And there are also evolutionary gene networks which function in the brain, and also, obviously, behaviors sometimes have an evolutionary track or trend to them. They're carried through from one species to the next. So the goals of neuroscience as a biologist sees it are to explain the mechanisms that process sensory information and direct the behavior of an animal. Right? There's sort of a statement of biological neurobiology. So for instance, for a simple response to a stimulus, as biologists we want to explain sensory transduction, that is the mechanism that converts the stimulus into an electrical signal. We want to understand perception. Now that's an interesting word, perception. Anybody want to define what perception is? Yeah.
How our brain, I guess,
Perceives things.
the information we receive from the sensory inputs.
How the brain, sorry. Too busy. Processes.
Yeah,
That's a bit mechanical, isn't it?
I guess.
I guess. Can somebody give me an example of the difference between perception and reality? Yeah.
Your eye, you have a blind spot that doesn't exist in reality.
Okay, yeah. In the eye you have a blind spot that does exist in reality, but you don't perceive it.
Something like that.
So there
isn't in the world a blind spot. You only see a blind.
Oh, you mean that there is, but you don't even see the blind spot.
Right, you see nothing. Like if there's maybe I was asked.
No, it's fine. It is it's the distance, right, between what is present in terms of the physics or mechanics of the process and what we actually experience.
I have
Yeah.
a better example.
Please.
If you put a dark color next to white, the white will look darker than it is. Or if you put a light color next to white, the white will look lighter than it is.
Everybody get that? If you put a dark color next to a light color, the dark will appear a lot darker than it is. And if you put the white, if you look at the white color, the white will appear whiter. In other words, there's contrast enhancement at the interface between the two colors. And yes, so that's an example of something that we perceive these differences, these subtle differences in the gradient of the colors at the interface between the two. But in reality, it's a sharp border with one kind of color on one side and one on the other. There's no mixing or enhancement. So we'll come to that, because that one has actually a fairly simple neurobiological explanation. Any others? Yes?
A mixture of hot and cold, maybe it's bring a lot of energy.
If you touch a mixture of hot and cold,
Yeah, so there's a rods and line.
Yeah.
Hot one's cold, and hot one's cold. Put your hand on it, and you're able to see this hot.
Yeah. Oh, okay. Mixture of hot and cold. So I'm going to put my hand on one hot rod.
Little rods. They're next to each other.
Two rods. Oh, I see. So I put my hand on two rods. One's hot and one's cold. And I never noticed the cold one. Yeah, I can see that. It's not part of my day-to-day experience. I don't see it.
I went to a kids museum. So like,
Oh, yeah, right. It's a kids museum, right? Yeah, that's where you see all these things. Any others? Let's just take one more. Yeah, from the back there with the great things like blank. You don't have those on. Appreciate that.
Would you like to consider watching a 3D movie?
Watching
Like a 3D movie?
a 3D movie. Yes. In other words, you see it as a whole. You experience it as an environment, not as an image that's being fed into one eye and a different image that's being fed into the other. Yeah, that's sort of a crude example of what's called the binding problem, whereby it's one of the mysteries of neurobiology is that we experience things as a gestalt, as a whole, whereas we know that what's coming in is coming in through different sensory modalities, like one eye versus the other. Actually, just going to the movies is a perceptual trick. Why is that? It doesn't have to be 3D. Why is that? What's the perceptual trick with the movies? Yeah.
Well, you see things that are that appear closer to you and further away from you, but really they're on the same plane.
That wasn't what I was thinking. The distance, the parallax.
Yeah.
Yeah, that's a good example of perception, where you process distance by looking at an object from different angles. But it's not what I was thinking of. Yeah. Next go.
You perceive that there are people or places on the screen, but it's not actually people or places, it's just really light in certain ways.
No. Never happens to me. Yeah, guy there. Yeah.
The film itself is images that are they're not actually moving images, but it's
Right.
a series of images.
Yes, right. So a film, at least I see digital films. I think they work this way. But old-fashioned film is much easier to explain, right? So it's a piece of celluloid, it's a film, and it has frames, right, like a cartoon. And each frame shows somebody in a different attitude, and they wind the film through the projector at a fast rate, and you see it as a continuous movement. Yeah, that is a perception. If you were a fly, you probably wouldn't see it as a continuous movement. You'd see it as a series of jerky movements. Why? Yeah.
Because the speed at which the images move is made specifically to work with how fast our brain can process those specific images.
And flies are,
They can probably process them a lot faster.
they can process faster. Actually, it's not their brains, they're photoreceptors. We're limited by the speed of this signal transduction. I bet in our photoreceptors. Okay. Oh, there's a whole line of text. Breaking every rule of writing lectures. So, the reason this is here, and you can go and read it later, is I wanted to read it out to you because it's really interesting. This is what a famous British neurophysiologist, said in 1953 about a problem that vexes us all in this room today, which is how can we sort of include the goal of neuroscience is to explore our minds, and yet our minds are doing the exploring. So can we ever really reconcile these concepts of consciousness and mind and perception with the crude physical description of how the nervous system and its information processing work? And this is something that worried him, that I think worries anybody who gets involved in neuroscience. And he has a very interesting thing to say about this problem. And so I'm going to read a little bit of it. There is, first of all, a fundamental difficulty which does not seem to me to be soluble at all by experimental science. The difficulty of connecting physical and mental events, of bringing sensations and thoughts into the framework that we use so effectively for describing our external world. All right, so this is the problem that I mentioned. I cannot myself see how further knowledge of brain physiology will help us to reconcile mind and matter. But I am prepared to believe that 100 years hence, it will be difficult to understand why we ever puzzled about it. Even now, there are many scientists and many philosophers who themselves find no difficulties and think they arise from faulty premises and outworn habits of thought. they may well be right. And what he does then is he brings, in the rest of the paragraph, an example of why they may be right. Why, in fact, there might be no difficulty in reconciling the description of the matter of the brain with the fact of the mind, the concept of the mind. And he does so by analogy. He mentions that in the early days of biology, there was a camp of biologists who believed that organisms derived from eggs. And there was another camp of biologists who believed that organisms derived from sperm. And they fought like crazy over this. So the notion that it might come from both together seemed too absurd to be considered until something happened to break the spell and the joint origin became a reasonable idea. So he's just using this as an example of how a biological revolution can sneak up on you and suddenly you don't understand what the fuss was about. And no doubt the Renaissance produced many examples of the same sort. Difficulties which vanished when ideas which had seemed inherent and unchangeable were found to be out of date. And so I am ready to believe that future generations will not find it as difficult as I do to include my mind in the scientific picture of the world. I'm content to think that it is a puzzle for me, but it may be no puzzle at all to someone without my particular preconceptions. And so you'll as a class, you'll fall on all sorts of levels of this gradient. I think I'm more with Adrian on this one. But the job here is to learn about the biology of the brain and not to have preconceptions about how to meld that with the concept of the mind. And to make your own level, make your own determinations, which will vary from one generation to the next. My historical perceptions are very different from Adrian's, and they're also very different from yours. So I can't help you with that reconciliation. I can teach you how the brain works, and we can discuss how the mind is involved. But what Adrian is saying here is that you have to do that yourselves internally. And it may not, for many of you, be a leap at all to do it. Okay. Mechanics. So let's start with some genes. When do I stop? 1:45? Is that when I stop? Oh, it's a long way away. Okay. All right. So these are the number of genes that are known to be in the genome. Now, what do we mean by genes? All those people who've got 330 or 207, what are they referring to here in terms of genes? Yeah.
Segments of DNA that code for
Specific traits? Specific proteins. I mean, people have varying levels of because often you can delete a gene and there'll be no phenotype, there'll be no actual result. So it's not coding for any trait that you can easily describe. But I guess the molecular, what you're giving in terms of traits is the sort of Mendelian view, and the molecular biologists here you all said proteins. All right, but I think this is what this refers to, is what they call structural genes, right? The genes that code for proteins, rather than all the bits in between those genes that determine where the protein is expressed, when it's expressed, what regulates its expression. So 20,000 in 20,000 in a human. C. elegans, little worm, nematode worm, has 19,000. And the point of showing this here is that we have a much more complex and better developed nervous system and can do all sorts of wonderful things with it that C. elegans cannot and does not. Right? So you can't find necessarily a connection between the gross number of genes in an organism and the complexity of its nervous system. Right? Now, one of the reasons for that might be that maybe as humans a lot of our genes are devoted specifically to expression in our nervous system. And C. elegans has to occupy its ecological niche. It's got to have different kinds of genes for different purposes. For instance, for olfaction maybe, or for motility in the soil. Right? So, because of the way evolution works to match an organism to its environment, it's perhaps not surprising that we are, in terms of gene numbers, we're as complex, or no less more complex than a nematode worm. We're all trying to survive in a particular environment. But anyway, despite that, you can't make that connection, right? So, what makes the human fly and mouse brain so different? So it's not the gross number of genes. They're not so different in mouse and humans. And relatively few genes, only a fraction of the genes, as we'll see, are specifically expressed in the nervous system in the brain. It's actually a number that increases all the time, but anyway, relatively few. So it's probable, therefore, that what makes our brains or nervous system different from those of the fly or the mouse is the spatial and temporal pattern of gene expression as controlled by introns and regions of DNA surrounding the actual structural genes themselves. Also, differences in the fine structure of certain proteins. So, for instance, we may have a dozen variations of a single channel because of alternative splicing of the RNA from that gene. Do you know what alternative splicing is? Yeah? Okay, somebody explain alternative splicing. What's alternative splicing? Yeah, give us a quick one sentence.
It's what you take out different exons to combine and, I guess, convert into amino acid chains to construct different protein.
Yeah, so a gene consists of introns and exons. And the exons contain the protein coding regions, and the introns don't. You know that? Okay, so then sometimes you have two alternative exons that code for the same part of the protein, but they're slightly different in their sequence. And what can happen is those two pieces of code can be alternatively spliced into the final protein. So you can have two flavors of protein. And some ion channels in the nervous systems, can have 30 or 40 different variations of alternative splicing. So alternative splicing. Alleles of genes, in other words, slightly different sequences for genes. And duplications of genes. And there's a very interesting paper that was published in Nature, I think last year, maybe, talking about how gene duplication during human evolution may have been associated with a big increase in brain capacity and complexity at the same time. So this is a situation where a single gene is duplicated, right? It's reproduced twice. And it's a very common mechanism of evolution. So some of those 20,000 genes may have duplicates, and the duplicates may be extremely important in driving brain complexity. So I put the link here to at least the News and Views article for the paper. The paper is a further link away. And you might want to, if you're interested in human evolution and genetics and brain structure, read it. The difference in anatomy also are driven by learning and environment. Right? So it's very important that you realize that the brain of any organism is not static in its structure or in its connections within it. It's driven by its environment and by learning that occurs. So all of these things are factors that ride on top of simply the gross number of genes that are expressed. So this shows you a sort of a Venn diagram of the genes. Of the 20,000 genes in the human genome, about 6,000 of them are expressed in the brain. So quite a number of them, but by no means all are expressed in the brain. Only in the brain. Then a further 8,000 are expressed both in the brain and in other tissues in the body. So they're uniformly expressed all over the body. And then there's another 6,000 that are not expressed in the brain at all. That's just some numbers. don't want to think of those 6,000 genes as being expressed necessarily only in one or two cells or groups of cells or a particular structure in the brain. All of these dots here represent the expression or a place in this brain where a particular gene, one gene, is expressed. And so you can see that all sorts of cells throughout the brain are expressing, making the protein for this particular gene, encoded by this particular gene. So genes are not necessarily highly localized in terms of their expression pattern. They can be switched on throughout the brain. And when you have a gene that is widespread in the way it's expressed, mutation of that gene can have a dramatic brain-wide effect. So this is a gene, the result of a mutation of a gene called ASPM. This is a gene which controls cell division. And not surprisingly, that gene is widespread in many cells throughout the brain. And also, perhaps not surprisingly, mutations can result in a widespread phenomenon. In this case, it's microencephaly. And so this is a patient with microencephaly with a particular mutation in that gene. And this is the image of the normal brain. So in the adult, or the same stage of development, the gene, because these patients tend to die somewhat young, so it's difficult to call this adult. But at the same stage of development, the brain is a lot smaller than this individual. And here is an individual with that, with this microencephaly. Here you can see the forehead circle. Okay. Now, by contrast, other genes are expressed in only a tiny part of the brain. So, although some genes are active in many cells, other genes can be only active in particular cells. And again, this shows a protein. It's actually a structural protein involved in connections between cells, called desmoplakin. And only this little band of cells in this small part of the brain here, the hippocampus, is actually actively expressing the gene, as shown by the fact that it lights up. Here's another gene, one that's involved in chromosome maintenance, like the one we saw before, that's widely expressed. Here are a couple. The lipoprotein lipase, a lipid metabolism gene, is only expressed again in a very thin layer of cells in that part of the brain. Yeah.
Is this a different presentation that you uploaded to Blackboard?
It is, yes, because as I mentioned, I think it says on the lecture that what happens is I sit down before the lecture and I go through my slides, and sometimes I think of things that would be neat, and so I put them in, or I see things that are wrong and put them in, or you see things wrong in lecture and put them in. So what I do is when I finish the lecture, I put off on a set of slides that said final. And that is the final one. So you can substitute these slides. Okay? And, I don't think there's any other way of doing it, because I don't want to be in a situation where I'm, writing my slides 4 days ago, putting them up, and then, just coming in and not even going through them again, trying to get excited about something I've just seen. Yeah. Okay, so what are the cellular components of the nervous system? And amazingly, these sort of very intricate patterns of expression of genes, as well as the very intricate structures of the brain, amazingly, are generated by only two basic types of cells: neurons and glia, right? Or neuroglia. And we'll talk about the structure and function of those now. But in some respects, the brain or the nervous system is a very simple organ. There's two kinds of cells, The liver or cardiac, the heart, the liver, kidneys, all have many different kinds of cells. But not these kinds. So this is your sort of basic neuron, right? It's got, in this case, it's got a cell body in the middle. At one end here is an extension called a dendrite. And at the other end is another extension, cellular extension, called the axon. And the dendrite has branches, dendritic branches at the top, which in this case have been reduced to these sort of novel little things, but sometimes, as we'll see, they're very beautiful branching structures. And the axon has a synaptic terminal here, which, again, here it's fairly compact and sort of novelly, again, it can be beautifully branched. Right? And so the plasma membrane of the cell covers all of these surfaces, right? These are all within the plasma membrane of the cell. So that's your sort of basic neuron. So the history of looking at these cells is kind of interesting. So here's our basic neuron. Here's the dendrite. Here's the axon. Here's the nerve terminal. Out here. For the longest time, when people looked at the nervous system, there's a nucleus. Get rid of the nucleus. So this again is the dendrite. This is the axon over here. The nerve terminal and the axon. Okay. For the longest time, when people in the 19th century, mid part of the 19th century, looked at nervous systems through microscopes, they thought that the neurons in the nervous system were all in continuous contact, cytoplasmic contact with one another. In other words, there was just a hole here where the two neurons were joined, where the nerve terminal of one neuron was joined onto the dendrites of the next neuron. They thought there was just basically a hole here, and cytoplasm could stream back and forth. And they called this the concept of the diffuse syncytium of cells, right? All joined together, syncytium. Interconnected neurocytoplasm. And so the neurons, in fact, were not quite distinct cells with surrounding membranes, all right? Because in this case, there's two nuclei and one membrane. There's not just one nuclei surrounded by a membrane. And so to that extent, they kind of violated the idea of the cell theory, the idea of the self-contained individual cells. All right. Now, even now, this actually seems like a very attractive idea compared to the reality of having separate cells. The reality of having no chalk. Separate cells, like that. That's the reality, right? We now know. So my question is, what makes the older idea so attractive? Yeah.
You wouldn't need neurotransmitters. You could just have the voltage-gated ion channels propagate the signal.
So he says you wouldn't need neurotransmitters, but you could have voltage-gated channels propagating the signal all the way down. That's absolutely correct. But you're arguing from a level, a sort of physics, a level of sort of biological detail that is a little bit higher than I would ideally like. You know, can somebody sort of go back to physics, basic physics, and explain this? I know you haven't had physics yet, but that doesn't mean you can't explain it to each other. So many of you have. So why is this attractive? I mean, so his answer is correct. But I'm trying to sort of deconstruct it into a, yeah.
It's a simplified structure of multiple cells, one full system of neurotransmitters when having just as data channels, but it's designed to what formal slide.
Well, it is simpler, I guess, because you don't have to have the neurotransmitters and the release, which is what he said. There was a big problem with your argument there because you talked about how the neurotransmitters could carry the signal sort of through the gap. Yeah? And that you just wouldn't need the neurotransmitters at all. Yeah?
It's a closed circuit, which...
A closed circuit.
Yeah, so
Like a motor racing circuit?
like, I mean, you said physics. So
Yeah, right, yeah. So what sort of circuit is it?
I mean, like electronic circuit.
It's a closed electronic circuit. Which one's closed?
The older version, the more attractive version.
And what do you mean by that?
I mean, it just, like, we end up electricity going through the circuit, and it's all connected, whereas when you bring it apart, then it's harder to grasp.
So where's the electricity flowing that you're thinking of? This way. Yeah? Right? That's the electricity you're thinking of. Right. Okay. Yes. That's correct. Right? Gets it down to a little bit more, because the problem is that if you have membrane there, does cell membrane let ions through, possibly a negatively charged ion? Without channels, just make it cell membrane. Is it a good conductor of electricity? No, it's not, right? It's made of fat. Right? Fat's not a good conductor, it's an insulator. Right? So without with the membrane between them, electricity can't easily pass from one cell to the next. Electrical current can't flow easily from one cell to the next, right? Okay, so now we can reconstruct what our friend said down here when I asked you, okay, so you say there's some sort of electrical current. Where does that electrical current come from? Where does the current that comprises that arrow? How's it flowing? Why is it flowing? Yeah.
Ion movement in and out of the neuron.
Yeah, it's coming from ion movement into the neuron. And what is that ion movement flowing through? Voltage-gated channels. Yeah, so there are voltage-gated channels. And we'll go through all this later, but I'm trying to get some vocabulary and figure out where you are. There are voltage-gated channels in the plasma membrane which are activated as the action potential comes down that neuron. And in the case of the syncytium, the current would simply enter the other cell. It would make the inside of that cell more positively charged and would trigger an action potential in the next cell. Right? Whereas in fact what happens is that the current actually just exits this cell through its own membrane. And what has to happen is neurotransmitter has to be released to trigger the action potential through a very complicated chain of events, which you will know in exhaustive detail by the end of the course, in the next segment. Okay. Now there are cells actually that work somewhat in this manner. There are connections in the brain which are made between neurons, which actually don't need neurotransmitter. And we'll come to those as we talk about particular parts of the brain. Right? But this is a much more attractive idea at the first glance for making a nervous system that runs on electrical signals. But it ain't so. And we can discuss for its great interest, why ain't it so? What did neurotransmitters buy? Okay, so the nail was driven into the coffin of this idea of a syncytium by a very simple invention of something called Golgi stain. So this was a cytological stain that stains neurons. It makes neurons black, actually in color. Specifically stains neurons. It doesn't stain glial cells much. And it was invented by Camillo Golgi in 1873. I believe Camillo Golgi was an Italian scientist. And it was extensively used by the turn of the century, before then, by a man by the name of Santiago Ramon y Cajal, who was a Spanish scientist. And Santiago Ramon y Cajal is sort of the indubitable father of neurobiology. To identify, and to use this, because what this stain does, it's kind of strange, and I'll show you a picture of it here. So this is a drawing, actually, of a section of the part of the brain, the cerebellum, I believe, which is stained with this Golgi stain. And as you can see, there are thousands and thousands and thousands of neurons in that slice, in that section. But the Golgi stain has only stained a few of them. It's only actually stained 2 neurons that are actually right in the section that's being made through the brain here, where you can see the whole thing. These guys here, you can't, and then there's some here of a slightly different structure, some here and here. But only 2 of this kind of neuron are stained. So the Golgi stain is really weird in that it sort of randomly stains neurons, just a few neurons in any one section. But the amazing thing about it is that it stains the whole neuron. And so this is sort of a refutation right there of the syncytium, because it's very difficult to understand. If the stain stains the whole of this neuron, why doesn't it spread into the next one if the cytoplasm is connected? So this was a puzzle that couldn't be addressed by the syncytium theory. And so Cahal proposed a number of things. He studied, his slides are still the most beautiful, really, of neuroanatomical slides available. He looked at hundreds of parts of the brain, basically defined the cellular anatomy of the brain. And so here he is. Here he is when he was sharp and in his youth, comparative youth. He'd already lost a lot of hair by then. And here he is in his old age. He doesn't look quite so sharp there. He looks a bit dull. There he is. And in case you wonder why did he have such an influence, you can see the resemblance. Every neuroscience class has to begin with a Monique Cahal slide, so there's yours. All right? A ritual that everybody has to go through. Okay. So he proposed 2 things. The principle of dynamic polarization. Information flows in a consistent way within each neuron from dendrites to the nerve terminal via the axon. So information flows from the dendrites through to the axon. So that's called the principle of dynamic polarization. And the principle of connectional specificity. There's no cytoplasmic connection between neurons. We now know that that is a general statement, that there are cytoplasmic connections between some. No cells do not form random networks. In other words, they just don't randomly join up with their neighbors to form a syncytium. They're highly targeted in terms of whom they make synaptic contact with. And each cell makes precise connections with other neurons via synapses, right? This is called a synapse, this connection between the nerve terminal of one cell and the dendrite of another, right? So there are about 10 to the 11 neurons in the human nervous system, and although they're all the same cell type, as it were, there's thousands of different structures, different subtypes. Each neuron can be contacted by thousands of synapses from other neurons. So these numbers give you some idea of the potential complexity of the brain as a computational network. In general, it's the same. Neurons have dendrites, cell body, an axon on the synaptic terminal. And the vast majority of neurons in the central nervous system are terminally differentiated and cannot undergo cell division. Many have a limited capacity for repair as well. And so this is a major problem in medicine. It means that damage to the central nervous system is often irreparable. So here's the flow of information through occipital neurons. So why does the information flow in this direction? It's because the dendrites contain receptors for neurotransmitters, NTs, neurotransmitters. That's in the case of a neuron that is stuck in the nervous system between 2 other neurons, like this one here. It's receiving information from another neuron. But there are also sensory neurons which receive stimulation from the outside world. Heat, touch, light, so on. And so the sensory neurons, they have sensory receptor proteins rather than neurotransmitter receptor proteins. Okay, so those are the 2 kinds of receptor proteins, and these are for sensory neurons only, so these guys here. So this is where the information impinges on the cell where it's generated within an individual neuron. And then the axon terminal makes synaptic contact, releases neurotransmitters onto the dendrites of other neurons, as we've seen here, or onto the dendrites of muscle cells, or indeed some endocrine cells as well, so as to cause muscles to move and glands to produce hormones. Okay, hence the adrenaline rush, for instance. Now this bewildering variety of different types of neurons, brave people, anatomists, have made attempts to sort of categorize neurons into varying types. And here, the way they characterize them is in terms of the number of processes that leave the cell body. So this would be a bipolar neuron, because it has two processes, one dendrite and one axon. This guy here is a monopolar neuron. These are monopolar neurons. They have a cell body with a nucleus. They have axons, but they have no dendrites. These are in the nucleus that contributes axons to cranial nerve 5. Let's look. This is again bipolar. Even though the axon branches, the dendrite branches up here, there are still two processes which leave the cell body. So this is another bipolar. This guy here is another monopolar cell. It has dendrites, but no axon. And this is a cell that's in the functions in the network of cells in your retina, of your eyes. Right, so there's monopolar cells and bipolar cells. And there's a question about these monopolar cells, the one before, which is, what's the flow of information? Right? Where do neurotransmitters act? On these guys here, where do the neurotransmitters act? Because there are no dendrites. Where are the neurotransmitter receptor proteins? And on this guy here, perhaps even more puzzling, where are the synapses? Where's the nerve terminal? How does this neuron signal anything to anybody else? Right? Can't have a neuron which doesn't signal anything. It wouldn't survive in evolution. Right? What's it doing? Signaling to itself? No, it's got to be doing something. So, the question is, to you guys, where are the neurotransmitter receptor proteins on this cell? Where do you think they are? Yeah.
On the cell body.
On the cell bodies. Yep. Anywhere else? Yeah. Yeah, there actually could be a few on the axons. Yeah, there could be. And what about this one? Where are the nerve terminals? Where is neurotransmitter being released from this cell onto some other cell that it's going to contact? Not the cell body. Which means the dendrites. Yes, it turns out that there are neurons which make contact or have processes, axons and things, and other dendrites that move through, that progress through this. They're not moving; they project through this dendritic tree here, and the actual dendrites make synaptic contacts with those with the other cells. And so you have to imagine that somewhere along in this dendritic network, there'll be little clusters of neurotransmitter vesicles. Neurotransmitter is being released there, and you have to imagine that other cells are projecting processes through here, either axons or dendrites, which this guy's contacting, right? Now these dendrites also have the neurotransmitter receptors in them. So you could imagine a situation where a cell that is sending an axon through here could be simultaneously making electrical contact with this network and then receiving neurotransmitter as well. And so now the idea comes that maybe this dendritic network is a very active processing unit that is both receiving information and processing information and outputting that information as neurotransmitter to alter the behavior of the stimulated neurons. Okay, so when you think of neurons, don't get stuck with Cajal's flow here. You also have to bear in mind that sometimes very complex little networks, processing networks, can exist in dendrites, or between one axon and the next, because there are no dendrites. And then there are multipolar cells. This cell, for instance, has a bunch of branching dendrites from the cell body. And this absolutely amazing dendritic tree here. So each of these branches has further branches and so on. This is the cell that Cajal was in that image of Cajal's. And you also see that here's a cell here, a cell body, that has multiple axons coming out of it as well through here. And those axons also have multiple branches themselves. And here's another little curlicue, right, of information processing. Here, the axon has a branch which comes back and contacts the dendrites of the same cell. Right, so stimulation of this cell sends action potentials down this axon, then back up this branch to contact the release neurotransmitter back on these dendrites. And so what you have here is a complex little feedback loop that's altering the information flow, altering that's processing information within this neuron itself as a single unit. So neurons don't have to contact one another to process information. They can do it sometimes all by themselves. Right? So lots of variations. I'm just going to run through the parts of the cell. I know this is boring for some of you because it's just recap of perhaps what you did in 230. Here's the cell body here. And here's the cell body as an electron micrograph, and here's the nucleus. And the important thing to remember about neurons is that the cell body here is the site of protein synthesis. There's a lot of discussion in the literature. Every now and again there's a little flare-up of discussion about whether proteins can be synthesized in the dendrites. Are there ribosomes in the dendrites that can synthesize proteins from messenger RNA that's transported up there? And there's some limited evidence, the last I heard was from Drosophila, that some neurons can do this. But nobody's terribly convinced. And so by and large, when you see a neuron, you should image the fact that protein synthesis is only occurring, probably, in the cell body, until you know otherwise. And I might do another, if you remind me on the discussion board, I might do my sort of annual literature search and see what the latest is on that. So if somebody reminds me on the discussion board, I promise to do that. But so far it hasn't been proven. The dendrites, so these are there's some dendrites, where is it? Yes, out here, they're branching, shown here on the electron micrograph. This, as we know, is where information is received by from nerve terminals, synapse. Then you have the dendrites, so the sites of the information transfer. Here are synapses being made onto a dendrite from another incoming axon from another neuron. Here are the neurotransmitter vesicles, the little vesicles that store neurotransmitter, and we'll release them onto the dendrite here. The dendrite is in purple, and these are in white, these nerve terminals. So here's this tiny cleft that exists between the nerve terminal and the dendrite. And the neurotransmitter just has to cross that cleft and excite the dendrite. We'll talk about that in detail later. Some of these axons here are myelinated. You see this axon here looks as though it looks like a string of sausages. And that's because it's wrapped in a fatty sheath called myelin, and we'll talk about the function of that later. But myelin is an insulator because it's basically a wrapping of membranes. And the myelin that wraps around some neuronal axons functions to speed up the action potential. And you notice that there are gaps here in the myelin sheath, and these are called nodes of Ranvier, whether we have that in there. Here's the node of Ranvier, the gap, where there's no myelin sheath. This is myelin, and a gap. So this is naked plasma membrane and myelin on either side. And you also notice that not every part of an axon is myelinated. The part that's just joining the cell body in this case is not myelinated. And that's where the action potential is often initiated, down here, not in the cell body, but in here, this part of the axon. Okay, I think you guys get 10 minutes to transfer. Is it? Yeah, 10 minutes to transfer, is that right? We normally finish 10 minutes before the end. Am I right about that? We run till 1:45? No, I'm not going to consider it. We finished. Right now, we've got a few slides to finish. But we'll finish now. I'll pick them up next time. I'm going to Manchester in England for the weekend. And my brother's birthday. So